Hello students, in this session I will talk on cultured media. So firstly, what is cultured media? Basically, culture is to maintain the growth and media is a nutritive substance or surface for the cultivation of organism. So a cultured media is a special medium which is used in microbiology laboratories to grow different kind of microorganism, and this is an artificial medium, so it needs a different nutritional component which is required for their growth. So the basic ingredient or constituent required for the growth in culture media include agar. Agar is the main component that is used universally for preparing the solid media. It is usually obtained from a seaweeds, and after necessary processing it is available in a powdered form, and it is usually used in a concentration of 2 to 3%, and this agar has a unique property of melting at 98 degrees centigrade and solidifying at 42 degrees centigrade, and they serve as a solidifying agent. And this agar also does not have any nutritive value and is also not affected by growth of bacteria. Secondly, we have peptone, which is another important ingredient of culture media, and this peptone is a complex mixture of partially digested protein. It is usually obtained by digestion of a lean meat or other protein materials such as the hard muscles, fibrin or soya flour with proteolytic enzymes such as pepsin, trypsin or papain, and it also serves as a source of nutrition for bacteria to grow. So this agar and peptone, they are the essential or the most important component of the culture media beside other ingredients. Other common ingredients of culture media include sodium chloride, buffers, indicators, selective agents, additive for enrichment, and reducing substances. Next, we have classification of cultured media. Here we have three types based on physical state, based on presence of molecular oxygen and reducing substances in the media, then based on the nutritional factors. So under this classification, again it is further subdivided. So the first one we have here is based on the physical states, and under the physical state we have liquid media, we have semi-solid media, we have solid media. So the first one, that is liquid media. This liquid media provide greater sensitivity for the isolation of small number of organisms, producing general turbidity, and in this liquid media no agar is added since agar has a unique property of solidifying. So the example of this liquid media we have nutrient broth. Next, we have semi-solid media. This semi-solid media is a media where the concentration of agar is reduced to 0.2 to 0.4% and makes the medium semi-solid. And also it facilitate the spread of bacteria in medium, and they are useful in demonstrating the bacterial motility and also separating the motile and non-motile strain. So in this semi-solid medium, less than 1% of agar is added, and the example we have here is motility medium. Thirdly, we have the solid media. So these solid media are somewhat less sensitive than liquid media, and they are mainly used in petri dish as plate culture, and sometimes in bottle or tube as tap or slope culture. And the main purpose of culturing in this solid media is to isolate a single colonies or discrete colonies. For example, we have Newton agar, which is one of the simplest and most commonly used medium in the laboratory. Next, we have based on the presence of molecular oxygen and reducing substances in the media. Here we have two types, aerobic media and anaerobic media. In aerobic media, this media cultivate only aerobic bacteria, which means the organism can survive and grow in oxygenated environment or in the presence of oxygen only. For example, mannitol salt agar. In contrast, anaerobic media are used for cultivation of anaerobic bacteria which grow in absence of oxygen. For example, we have CMB, and it is also known as the RCM. Next, we have based on nutritional factors. So under the nutritional factor, the first one we have here is a simple media. So this simple media support the growth of microorganisms that do not have special nutritional requirement. For example, we have nutrient broth and nutrient agar, and this nutrient broth and nutrient agar, they are used routinely for isolation of many bacteria from the clinical sample or the specimen, and they are also used in preparing the enriched media and for subculturing pathogens from differential or selective media for biochemical or serological testing. Next, we have synthetic media. This synthetic media is also known as the defined media, and they are prepared from pure chemicals, and the exact composition of the medium is known, and they are used for special studies and research purpose only. Here, Dubos medium with twin 80 is an example of the synthetic medium. Non-synthetic media. Most of the media other than basal media are usually known as complex media, and these complex media have some complex ingredients which consist of a mixture of many chemicals in unknown proportion. For example, we have chocolate agar, McConkey agar, RCM, and LG medium. Next, we have here is a special media. So this under the special media, we have different media. The first one we have here is an enriched media. The enriched media are invariably solid media, and they facilitate the growth of certain fastidious bacteria. These medium are prepared by adding substances like blood or serum to the basal media in order to meet the nutritional requirement. For example, we have a blood agar and Loeffler serum slop. Next, we have enrichment media. So this enrichment media, they are liquid media, and they have a stimulating effect on the bacteria to grow or inhibit its competitors, which increase the number of wanted bacteria in relation to other bacteria. For example, we have here selenite F broth and tetrathionate broth, which are used for the isolation of Salmonella typhi and Shigella. Next, we have the selective media. So this selective media are solid media that contain substances to inhibit the growth of one organism and facilitate isolation of a particular or a certain bacteria. Example, we have TCBS agar. So this TCBS agar is used for isolating the Vibrio cholerae from fecal specimen to inhibit the growth of another bacteria. Next, we have here differential media. So this differential media distinguish one microorganism from another growing on the same media by the growth characteristics. We have an example of McConkey agar. So in McConkey agar, the bacteria which produce lactose fermenters, they produce the pink color colonies, whereas those bacteria which are non-lactose fermenters produce pale colorless colonies, as you can see in the image given. So by producing these different colors based on the organism, it is known as the differential media. Next, we have indicator media. So this indicator media, they contain an indicator, and it helps to distinguish one organism from another, which changes color when a bacterium grows on the same media. Example, McConkey agar, which produce NLF and blood agar, which produce different type of hemolysis such as alpha, beta, and gamma. So with the help of different type of hemolysis, it helps to indicate the organism by their growth characteristics. Last but not the least, we have a transport media. So this transport media, they are used in case of delicate organisms. Example, gonococci. Since it Since this transport media maintain the viability of certain delicate organism in clinical specimen during the transport to the laboratory, because sometimes some organisms like gonococcus may not survive the time taken for transit or may be overgrown by non-pathogenic bacteria like cholera organism. So the example for transport media we have here is a steward transport medium. So, what is the use or what is the purpose of this culture medium is that it is important to grow microorganism outside the body as to identify the cause of infection from the clinical sample or specimen, so that proper treatment can be given, and also to study the characteristics or properties of different microorganism. So that's all for today, and thank you.